We are back with the diagram. And so over at the left-hand side under translation, this is talking you through the process. And over on the right, or actually toward the middle, you can see this is a nice graphic representation of the process. So normally, translation is dependent on having that cap at the 5' end because factor 4e binds it, and the ribosome will not start translating unless factor 4e has bound and locked factor 4g into position and locked all those other things down. It just has to make contact with every part of that. Otherwise, no translation. But what the virus does is, one, it goes in and it cuts factor 4G. It just cuts the end off it that would normally bind to factor 4E, which is a rotten thing to do. And then it folds up into that internal ribosome entry site, IRES, which is the sort of squiggly line shown down here at the bottom, right there. And that is going to simulate. That's going to make the same sort of contact with a ribosome as EIF4E bound to a cap made. And so that is going to be enough. The ribosome will dutifully translate whatever it comes after, which is going to be picornavirus. And here you can see some of the other parts. Factor 4A is shown here. This is that helicase. And here's factor 4E, and there's the 4DS, so the small ribosomal subunit. And once this all locks into place at the AUG initiation codon, the start codon, then the large subunit, which is going to be called 60S, comes along, and that will actually start the translation. So that's the point at which we actually start making protein. Yeah. So, oh, is that the end? Oh, no, it's not the end. Okay. So, but like I said, picornaviruses are very good at shutting off host translation. So you can see right over here. So this is the normal size of factor 4G on a gel. So we're running the gel, and big things will end up at the top, and little things will end up at the bottom. So, all right. So mock means one that is not infected, and so you can see this is how big 4G is supposed to look like, and you just get this extra little band there in the middle. That doesn't mean anything. That's just a thing. It's an artifact of the process. So after picornavirus infection, you got different times here, and so look at 2 hours after, you've got about half of your factor 4G still there, and half of this shorter thing that's binding to the same antibody as 4G. So it's 4G, but it's a cleavage product. It's the little piece that's left from factor 4G. By 3 hours, look at that. We've got tons of the cleaved version, so the cut version. Look how much factor 4G is left in the entire cell. It's something like 5% of the total. And then by the time you get to 4 hours, 5 hours, 6 hours, there is absolutely no factor 4G left in the cell. And with no 4G, there can be no more cap-dependent translation. And so the virus has won, and the cell is dead but doesn't know it yet. Yeah. And so this is what it does to translation overall. Over on this side, here you can see the mock translation, or rather mock infection, so not infection. And you can see, yeah, these are all the proteins that the cell is trying to make, separated out into a gel, and there's something like 20,000 of them. But you can see the really high copy number proteins, the really common ones, make the darkest bands. And you can see after 1 hour of infection over here, it pretty much looks like that. That's pretty good. But now look what happens from 2 hours. Whoa. Look at that just thin out. See how we're losing a lot of those bands that we had at 1 hour? And remember, 2 hours is the point right over here where we have cleaved about half of the EIF4G. And then look at 3 hours. 3 hours, we're pretty much just making this set of proteins. And you can see we pretty much lost all of our factor 4G. These are the picornavirus proteins. This is the big polyprotein, and these are all the little cleavage products of the polyprotein and all the sort of intermediates where we've got one part but not yet another part. So we've gone from a cell that was making this many proteins here, like enough to stay alive, over the course of 2 hours, between 1 hour and 3 hours, we've shifted over to a cell that exclusively makes picornavirus protein on demand. It's like, a crazy Amazon fulfillment center kind of thing. It's just super focused on one task and ridiculously efficient. Now, the other thing you'll probably notice is that, oh, look, here at 6 hours, everything starts to fade out. And by 7 hours, it's pretty much gone after that. The cell dies and explodes at about 6 hours. So that's where all the protein's going. There are pretty much no cells left that are still alive on the dish to give us protein, and that's what you're seeing there. So yeah, it is a real rocket ride to destruction. That's the picornavirus infection cycle. This one is poliovirus, one of those picornaviruses, but you could show the same sort of gel for any other picornavirus. They all do the same thing. They all have the same effect on the ribosome, all the common ones anyway, in people. Yeah, there's a lot of different picornaviruses, but let's focus on this. So we're going to pretend poliovirus is our model picornavirus, and then if you go on to study picornaviruses someday, and that's your thing, then fine. You can find all the all the exceptions to the rule, if you like. Okay, that is Picornavirus, and shutting off host translation and turning on virus translation. And you can see it is really, really effective, very efficient.